Welcome student to the third part of chapter 14, that is respiration in plants. TCA cycle, which is the Krebs cycle. TCA cycle, the full form of TCA cycle is tricarboxylic acid cycle or the Krebs cycle or citric acid cycle, was first elucidated by Hans Krebs. And that's why the name of this cycle is also Krebs cycle. The steps for TCA are: it begins with condensation of acetyl-CoA, that is acetyl group with oxaloacetic acid to form citric acid in presence of an enzyme which is citrate synthetase enzyme. A CoA molecule here is released out. The next is citrate is isomerized to isocitrate. The third step is decarboxylation of isocitrate to alpha ketogluteric acid. The decarboxylation of alpha ketogluteric acid will further move ahead with decarboxylation of alpha ketogluteric acid to form succinyl CoA. During this course of reaction, carbon dioxide and NADH is formed. Also, one molecule of GTP is formed, which will later convert into ATP. The fifth step is succinyl CoA is converted to succinic acid and a GTPA molecule is synthesized. In a coupled reaction, the GTP is converted into GDP with simultaneous synthesis of ATP from ADP. The oxidation of succinate to fumarate and then to malate is the next set of reactions. And finally, malic acid get oxidized to form oxaloacetic acid. At 3 points of TCA cycle, NAD plus is reduced to NADH plus, H plus. At one point, FAD plus is reduced to give FADH2. Continued oxidation of acetyl-CoA via TCA cycle requires continued replenishment of oxaloacetic acid. It also requires regeneration of NAD plus and FAD plus from NADH and FADH2. So the overall or the summary of Krebs cycle equation is 1 molecule of pyruvate plus 4 NAD plus 2 H2O plus ADP plus Pi inside the mitochondrial matrix will give 3 carbon dioxide plus 4 NADH plus 4H plus FADH2 and ATP. Thus, a glucose is broken down to give 6 molecules of carbon dioxide, 8 molecules of NADH, 2 molecules of FADH2 and 2 molecules of ATP. Here on your screen, you can see a Krebs cycle in detail along with the enzyme. So we will concentrate here the enzyme. In the first reaction, the enzyme which is involved is citrate synthetase. In the second, it is aconitase. When the isocitrate converts into alpha ketoglutarate, the enzyme involved is isocitrate dehydrogenase. The conversion of alpha keto to succinyl-CoA is done by alpha ketoglutarate dehydrogenase. The conversion of succinyl-CoA to succinate is performed by succinyl-CoA synthetase. Conversion of succinic acid to fumaric acid is done by succinic dehydrogenase. Then with the help of fumarate is converted into malate, and by the action of enzyme malate dehydrogenase, malic acid is converted into oxaloacetic acid. So this is overall reaction of Krebs cycle. Also, conversion of acetyl-CoA, sorry, it's formation of acetyl-CoA from pyruvate. It's done by pyruvate dehydrogenase enzyme. Lastly, once again, entire Krebs cycle. Oxaloacetic acid gets converted into citric acid by reacting with acetyl-CoA. Citric acid then converts into cis-acotinate, isocitrate, and oxalosuccinate. Oxalosuccinate will convert into alpha-ketoglutarate, to succinyl-CoA, and then to succinate. Succinate will form to fumarate, finally malate, and malate gets converted into oxaloacetic acid. The enzymes are citric synthetase, acotinase, aconitase, isocitrate dehydrogenase, isocitrate dehydrogenase, alpha keto glutarate dehydrogenase, succinate thiokinase, succinate dehydrogenase, fumarate, and malate dehydrogenase. So this is what the overall reaction, overall steps of Krebs cycle. Thank you for your attention.